Hello, everyone, and thank you for this wonderful opportunity to speak of my research in the InterRidge webinar series. And as Professor Lee introduced, I'm Min Sup Shim, an Associate Professor of Geomicrobiology in Seoul National University. The title of my talk is that the sulfur isotope as a qualitative and potentially quantitative tracers of past microbial activity. Here are topics of my presentation. First, why microorganism is important in Earth system, and the significance of microbial isotope fractionation as a geochemical trace fossils, and what we can learn from a wide range of microbial sulfur isotope fractionation in nature. Actually, most of this topic is not directly related to the mid-ocean ridge system, but I hope it can help you to utilize the stable isotope to trace the microbial activity in the ridge and other plate binary systems. I will start this lecture with a brief overview of why our scientists care about the microbes. First of all, the microbes is an important geochemical agent. In this slide, you can see the plant and animal and microbes. We know that what plant and animals do. Plants do the oxygen photosynthesis, and they fix the CO2 to the organic matter using the radiation energy from the sunlight. And animals do the exactly opposite reaction. They oxidize the organic matter to CO2 and reducing the oxygen to the water. So in this equation, we can realize that the plant and animals should be the important component of the global oxygen and carbon cycling. So what microbes can do is they can do the same thing. They can do the oxygen photosynthesis, and they can also do the aerobic respiration, but they can do a lot more. For respiration, they can use the nitrate and sulfate and solid iron and the toxic perchlorate and many other oxidants for the respiration as an electron acceptor instead of oxygen. It means that microbes can be involved in the variety of global elemental cycling, including the nitrogen and sulfur and iron and chlorine. Moreover, they can use chemical energy to fix the CO2 to organic matter. It means that microbial ecosystem can thrive even in the absence of the sunlight. And they can survive in the extreme environment. They can live up to the several kilometers below the Earth's surface, and they can live in the water 10 times more saltier than the seawater. And they can survive at temperature higher than 100 Celsius degrees and below than zero temperature. So it means that microbes carry out the geochemically important reaction almost everywhere of the Earth's surface environment. So now we know that the microbial component is an important component in the Earth system these days, but also they have a long evolutionary history. They appeared on Earth 4 billion years ago, and after that, the first half of the life history is belong to the microbial organisms. So if you want to understand the history of our planet, we have to study that interaction between the microbes and our system over geologic timescale. And how we can do that? Here is modern microorganisms and their remains. If you want to understand what microbial organisms can do in modern environment and your laboratory cultures, we can use a variety of molecular techniques. We can use DNA, or we can use protein, or we can use RNA. But most of these molecules cannot survive at the elevated temperature for a long time. For example, DNA in the geological materials can go back to the few million years, but it is so young when you think about the 4 billion years history of the Earth and life evolution. So we need something can persist for a long time in the geological materials, even after all the molecular and biochemical evidence is disappeared. There is 3 possible ways to do that. First, we can use the fossils, just like when we study the dinosaurs. This is a stromatolite, organo-sedimentary structure produced by the microbial community, and sometimes we can find the fossils of tiny microbes, something like this. But many of the microorganisms cannot leave physical remains. Even though they cannot leave any physical evidence, sometimes they can leave the chemical evidence of life. If the sum of the organic matter is produced by microbial activity and can be preserved in the geological materials for a long time, and this material is produced only by a specific group of organisms, we can use this molecule as a chemical fossils. We call this as a biomarker. And this is something like a geochemical body fossil of microbes. If biomarker is a body fossil, we can think about the trace fossil of microbial activity. Indeed, we can use that stabilized ratio as a trace fossil of the microbial activity. But how we can do that? The simplest answer is that microbes like using light isotopes. It is true, but it is also true that many of the chemical reactions also have a preference to the light isotopes. So we have a clear theoretical ground to support the biogenicity of the certain isofractionation. Here I show you one example. So this is the reaction of AB is dissociated to the A plus B. As you can see in this reaction coordination diagram, the energy state of AB is higher than the A plus B. It means that this dissociation reaction can happen and release some energy. For biological reaction, this energy can be used for the growth or maintenance. But actual reaction is a little bit complicated than this one, because the transition state between the reactant and product usually unstable and have a higher energy. So you can see the activation energy barrier between these two, reactant and product. It means that eventually this reaction releases some energy, but if you want to push this reaction forward, you should spend additional energy to overcome this activation energy barrier. Then now we think about the isotopes. So if you have two isotopes, then what kind of change we can expect in this diagram? Now we can see the energy change in the light isotope molecule and heavy isotope molecules. Usually the bond between the heavy isotope have a more stable than the bond between the light isotope. It means that they have a lower energy state. You can see this pattern in this diagram. And also this energy change makes the changes in the activation energy barrier. You can see in the light isotope, the height of the activation energy barrier is lower than that of the heavy isotope. That means that molecules containing the light isotope can participate in this reaction in the faster rate. So the more of the light isotope can be accumulated in the product side. So this is the kinetic isotope effect. But so far, I didn't talk about the biology at all. So in the purely chemical reaction, we can expect the isofractionation and the light isotope can be accumulated in the product side. But let's see what is actually happening in this diagram. In many of the chemical reactions utilized by microbial metabolism have a very high activation energy barrier. So in the purely chemical reaction, this reaction is usually very difficult to happen. So because of the high activation energy barrier, we should have given them an infinite time if you want to see that this reaction is actually happening. So at this point, we should think about the role of the biology. As for me, biology is a kind of a catalyst. So they reduce the size of the activation energy and accelerate the overall reaction. Something like that. Now we will see what happened in the presence of biology. Now this reaction can happen. But still, we can see the difference in the activation energy barrier. For the light isotope, activation energy barrier is lower when you compare that activation energy barrier for the heavy isotope. So in this situation, light isotope can be accumulated in the product side. This is an important contribution of biology. They make the room for the isofractionation to be expressed. Isofractionation itself is a purely physical chemical process, not specific for the biology, but In the absence of biology, many of the isotope fractionation process cannot happen within the regional timescale at the Earth surface environment. So that is the reason why we can use some of the isotope signature as a strong evidence of the microbial activity. Now I want to move the sulfur story. So this is the green lake in the New York State in the USA. This is a little bit weird lake below the certain depths. And water column is anoxic, and it contains the sulfate and sulfide. And the sulfate and sulfide have very different isotope composition. As you can see in this equation, higher delta value means that more of the heavy isotope, and lower delta value means that more of the light isotope. So you can see in this figure, sulfate in this lake have the sulfide composition of plus 20 per mil, and sulfide have the minus 30 per mil. It means that sulfate is isotopically heavier than the sulfide. Actually, this fraction is made by the sulfate reducing bacteria activity. Not only for the green lake, we can see the similar pattern in other places. This figure shows that the sulfide fractionation between the sulfate and sulfide reported in previous environmental and pure culture studies. In every studies, you can see that the isotope composition of sulfate is heavier than the sulfide. It means that sulfate reducer present everywhere in the anoxic aquatic environment. But if we want to trace back the deep past, we have some kind of archive of sedimentary sulfate and sulfide. Fortunately, we have that. In this figure, you can see that the sulfate can be preserved in the rock record as evaporated minerals or impurity in the carbonate rocks. And sulfide can be preserved as a pyrite or organic sulfur compound. So we can see that we can measure the isotope composition of sulfide and sulfate in ancient aquatic system. So this is the sulfide composition of sedimentary sulfate and sulfide over geologic timescale. You can see the sulfide is generally isotopically lighter than the sulfate. And even in the 3.5 billion rocks, you can see the fractionation. That means that microbial sulfate reduction is one of the oldest metabolism we can identify in the geochemical record. Now we see the signature of the microbial activity in terms of sulfide fractionation in modern and ancient environment. But there is one more interesting thing here. Wide range of fractionation. You can see the range of fractionation up to 80 per mil. It means that if you can understand that what control the size of fractionation, we can get much more information from the sedimentary or environmental sulfide subsignatures. Actually, if you see the pure culture studies using the sulfate reducing bacteria, you can already see the fractionation up to 70 per mil. So it means that most of the fractionation can be explained by the activity of sulfate reducer. But not only for this process, and there is another process involved in the sulfide subsignatures, including the diagenesis or some thermochemical reactions. But at this point, I just want to focus on the sulfide fractionation produced by the microbial sulfate reduction. Here is sulfate reducing bacteria. So they reduce the sulfate to sulfide. So sulfate is the main substrate for their metabolism. And we can expect that sulfate can do something for the sulfide fractionation. Actually, the sulfate limitation decreases the size of the fractionation. In this figure, you can see that below the 200 micromole of sulfate, sulfide fractionation is just a few per mil. So it is quite small fractionation. But I want to stress that sulfate reduction is not a half reaction. This is the redox reaction. So if you want to reduce sulfate to the sulfide, you should oxidize something. So usually they oxidize organic matter to CO2. So this organic compound, organic electron donor, may have some impact on the size of the fractionation. In this diagram, X axis shows that the rate of the sulfate reduction, and Y axis shows that the size of the fractionation. And you can see the electron donor limitation increase the size of fractionation, but decrease the rate of the sulfate reduction. So why the availability of the sulfate electron acceptor and the electron donor impact so differently on the sulfide fractionation? To answer this question, I will take one step deeper inside of the cell. This is the growth curve of the desulfur alaskansis sulfate reducer using the lactate as an electron donor. Usually, they produce a very small sulfide fractionation, just a few point fractionation. But if you look at the cell at the exponential growth phase, you will see the intracellular sulfate is very heavy compared to the sulfate outside of the cell. You can see the difference of the 50 per mil. Actually, we could measure the alpha sulfide composition of intracellular sulfate using the multi-collector ICP-MS techniques. But when cells enter the stationary phase, at this point, they consumed most of the lactate, but still the plenty of sulfate is remain in the medium. Then you will see the sulfistopic difference between the sulfate in and outside of the cell is small. You can see just 4 per mil fractionation. This striking difference gives us some hint why the sulfide fractionation vary depending on the physiology. So this is the cell of the alaskansis, and this is sulfate in the medium, and this vertical position approximates the sulfide composition. To reduce the sulfate to the sulfide, they first uptake the sulfide inside of the cell. So this is uptake step. During this step, no sulfur-oxygen bond breaks. That means that there should be no major isofractionation. Once they uptake the sulfate into the cell, they reduce sulfate to the sulfide using some enzymes. During this process, isofractionation happens. But the transported sulfate, not all the transported sulfate is reduced to the sulfide. Some of them leak out of the cell, something like this. So this is how sulfur flow inside the cell during the sulfate reduction. Or maybe some of you noticed that something is wrong in this diagram. Mass balance doesn't work. In this diagram, you can see the one influx into the cell and two outflux from the cell. We don't know the exact size of each flux, but the average sulfide composition of these two outflux does not match with the isocomposition of this uptake flow. How can you fix that? Because the sulfate reduction utilizes the light isotope faster, so the remaining sulfate inside of the cell should be isotopically heavier than the sulfate out of the cell. Then we can see that the sulfide fractionation at the cellular scale can be smaller than the sulfide fractionation actually produced by the enzyme inside of the cell. Here you can see the two end members. This end member, sulfate reduction is so fast, and only the small portion of the sulfate leak out of the cell. In that case, the isocomposition of uptake sulfate and the produced sulfide should be almost the same. But another end member, only the small portion of the sulfate is reduced to the sulfide. Then most of the sulfate just leak out of the cell. So in that case, the isofractionation made by enzyme can be fully expressed at the cellular scale. And the next question is that what controls this ratio of the metabolic fluxes? Again, I want to say that the sulfate reduction is not a half reaction. We need electrons. So the balance between the sulfide rate of the electron acceptor and electron donor fundamentally controls the ratio of the sulfate being reduced and leak out of the cell. So if you have the plenty of electrons, sulfate reducer can push the sulfate reduction forward, and most of the sulfate can be reduced. We can expect a smaller fractionation, but they are limited by electron. They can reduce only the small portion of the sulfate to the sulfide. Then many of the sulfate should leak out of the cell, and we can expect a larger fractionation. So this framework can explain that why the balance between the organic electron donor and the sulfate control the sulfide fractionation in the previous pure culture studies. With this qualitative framework, Now I want to move to the quantitative model of the microbial sulfide fractionation. In the previous slide, I only talk about the reversibility of the sulfate uptake step, but multiple reversible steps are involved in the microbial sulfate reduction inside of the cell. In the 1973, Rees proposed the linear network model for the variation of microbial sulfide fractionation with multiple reversible steps. This model has been updated several times to reflect the new experimental observation, but essential part is same. Overall fractionation is controlled by the isofractionation made by each enzymes and the reversibility of each enzymatic reactions. So if we want to test this model in the quantitative manner, we should know that the isofractionation imparted by each enzyme, and also should know what control the reversibility of each reaction in the microbial sulfate reduction pathway. I will talk about the enzyme first. Here are 4 major enzymes in the microbial sulfate reduction pathway: sulfate permease, and ATP sulfurylase, and APS reductase, and sulfite reductase. Sulfate permease uptakes sulfate inside of the cell, and ATP sulfurylase activates sulfate to APS using ATP to ease the following reductive reaction. And the APS reductase reduces APS to sulfite. So this is a 2-electron reduction. And sulfite reductase reduces sulfite to the sulfite. So this is a 6-electron reduction. In the previous models, assume that these 2 reductive steps produce the significant isofractionation, and all other steps produce the negligible isofractionation when compared to these 2 reductive steps. And all these fractionation factors define the range of fractionation made by microbial activity. Until recently, but the isofractionation by these enzymes has not been determined in experimental studies. In 2015, Will Levy studied the DSR-AB and measured the fractionation during the reduction of sulfite to the zero-valent sulfur by DSR-AB. He reported the 15 fermi fractionation. Although it is not the complete reduction to the sulfide, the 15 fermi fractionation is much smaller than the 50 fermi fractionation previously assumed in the models. I also studied some enzymes, and I studied APS reductase, and I measured the sulfide fractionation during the reduction of APS to sulfite. I got 20 fermi. So this is more consistent with the previous model prediction. Next is the reversibility. So as we talked before, for the sulfate reduction, these bacteria need some sulfur metabolite and enzymes, and also reduced electron carriers and ATP. So the concentration of all these materials control the free energy change of each enzymatic reaction, thereby control the reversibility of each reaction and the isofractionation. But not many studies actually experimentally investigated these variables. But our ongoing experiments show that the concentration of sulfur metabolite and the intracellular phosphorylation and redox potential can change depending on the growth stages and environmental conditions. So probably we will soon understand how this biochemical process can translate the environmental parameters to the fractionated ratio of the sulfide isotopes. Our quantitative understanding is not complete at this point, but I want to show you that how this approach can help us to interpret the sulfide composition in nature. Here is one example. So in this cartoon, you can see the first 2 steps of the sulfate reduction is reversible, and later 2 reductive step is irreversible, probably due to the fast supply rate of the electrons. In this situation, we can expect that the isofractionation by APS reductase can be expressed because some of the APS converted back to the sulfate. So this is the branching point, and we can expect some isofractionation. But for the sulfate, every produced sulfate should be reduced to sulfide. So in this quantitative reduction, we cannot expect any isofractionation. So the maximum fractionation in this situation should be the 20 per mil. But there is another way to access the reversibility as a sulfite step. We can use the oxygen isotope. Because sulfide can exchange oxygen isotope with water, but sulfate cannot exchange oxygen isotope with water. So in this situation, we can only expect the oxygen isotope exchange between water and sulfide, not water and sulfate. Interestingly, in the experiment done by Bruno in 2012, we can find the experimental result exactly consistent with our expectation. In this diagram, y-axis have a very complicated name, but it just indicates that the extent of oxygen isotope exchange between the water and sulfate. As you can see in this diagram, sulfur isotope fractionation smaller than 20 per mil severely limit the oxygen isotope exchange between sulfate and water. Now the cells are a little bit more limited by the electron flow. Then the sulfide can be reoxidized back to APS and sulfate. Then we can expect a larger isotope fractionation because the isotope fractionation imparted by DSR can be expressed at the cellular scale. But also we can expect the isotope exchange between water and sulfate because the sulfide can be reoxidized back to sulfate. This is consistent with the Bruno's experiment result. In this figure, in the case of rapid oxygen isotope exchange, so this data plot, you can see the isotope fractionation is greater than 20 per mil. So this is some consistency between the oxygen isotope experiment and our quantitative model expectation. I want to apply this framework to understand that ancient sulfur isotope record. This is isotope composition of sedimentary sulfide and sulfate over geologic timescale. And you can see during the Archean period, isotope fractionation between the sulfate and sulfide do not exceed the 20 per mil. So this 20 per mil is the maximum fractionation in the Archean sulfur isotope data. And this is the same as the isotope fractionation made by the APS reductase. Maybe this is just coincidence or maybe not. So I hypothesize that electron donor may be responsible for this pattern. As we talked before, if the cell have plenty of electron and the sulfide cannot be reoxidized back to sulfate, the maximum fractionation should be 20 per mil. Perhaps in the Archean ocean, the concentration or reactivity of organic compound were higher than the modern ocean, but in the absence of aerobic heterotroph, sulfate reducer might have the better access to the labile organic compound in the Archean ocean. In any case, it is very likely that the Archean sulfate reducer maintain the intracellular redox potential negative enough to prevent the reoxidation of sulfite to sulfate. So far, I have talked about the variation in the sulfide fractionation during the microbial sulfate reduction. But in the earlier part of this presentation, I also mentioned that there is other process can impact the sulfide signature in nature. For example, other microbial process and some chemical reactions. So all these reactions is involved in the sulfide fractionation. One may think that so many information is condensed into the single sulfide value, which cannot be properly deconvoluted. Actually, that is true. In some cases, two completely different reactions give you the exactly same sulfide composition. And we need more information. Fortunately, sulfur have the 4 stable isotope. We can find more information there. But traditionally, sulfur isotope geochemists only use the 32-34 fractionation because of the mass-dependent nature of the sulfur isotope fractionation. If you collect a random material and analyze their multiple sulfur isotope composition, you will probably see that this old isotope composition plotted along this mass-dependent fractionation line. Defined as the delta 33 sulfur is 0.515 times of delta 34 sulfur, and delta 33.6 is 1.9 times of delta 34 sulfur. If that is true, this minor iso composition cannot give you any additional information. But in the strict sense, you should see the very tiny deviation from this mass-dependent fractionation line. Because this mass-dependent relationship is defined as this exponent, and the value of this exponent changes slightly depending on the reaction and the chemical species. So we define this deviation as a cap delta 33 sulfur and cap delta 36 sulfur. Advances in the sulfide geochemistry for the last few decades made it possible to use the full 3 dimension of the sulfide subsystem. Here is a classical example of how the multiple sulfide subsystem help us to interpret the sulfide subsignatures. In this figure, X actually shows that the 34 fractionation during the microbial process, fractionation between the sulfate and sulfide, and Y actually shows that the ratio between the 33 fractionation and the 34 fractionation. Here you can see 2 metabolic pathways, sulfate reduction and the sulfur disproportionation. Sulfur disproportionators can split the elemental sulfur to sulfate and sulfide and get energy from this reaction. But if you just measure the 34 and 32 ratio of the sulfate and sulfide, as you can see in this diagram, it is very difficult to distinguish the sulfate reduction from the sulfur disproportionation. But if you have the 2-dimensional data set, you can see these 2 microbial processes have a distinct 33 fractionation pattern. So it is very easy to distinguish this microbial process in this 2-dimensional plot. Recently, I set up the experiment for the multiple sulfide analysis in my lab and analyzed some samples from the hydrosomal band in Indian mid-ocean ridge. But here I want to show you that the more simple example. I just collect the pyrite from the uraniferous slate in the southern part of the Korean peninsula and analyze their multiple sulfides composition. In this figure, you can see that the ratio between the cap delta 33 and cap delta 36, and the ratio is at about -8. This is consistent with the mass-dependent fractionation pattern. And in this figure, you can see the delta 34 sulfur of this pyrite is relatively heavy, up to 16 per mil. So it is likely that the magmatic process did not provide the sulfur for, did not act as a sulfur source for this pyrite formation. Probably this pyrite formed by the reduction of the seawater sulfate. But based on the 34 sulfides composition, it is not clear if this reduction is made by the thermochemical reduction or biological process. But the wide range of cap delta 33 value indicate that potential microbial sulfur cycling. So here I show that just 2 simple examples of the usefulness of the cap delta 33 to understand the microbial sulfide fractionation and the sulfide subsignature in the ancient rock record. But we still have one more term, cap delta 36. So if you have more complicated sulfur cycling problem, then you can use that the 3-dimensional plot to solve that equation. Here are summary and implications. First, strongly fractionated isolation can be geochemical fingerprint of past and ongoing microbial activity in extreme environment and billion years old rocks and extraterrestrial environment. Second, extensive experimental and theoretical studies demonstrate that the microbial physiology systematically controls the variation of microbial sulfide fractionation, providing a general model for the dissimilatory isotope effect. It can inspire the physiological investigation of other microbial isotope fractionation process. Finally, the analysis of all 3 isotope ratios of sulfur have provided an additional dimension to distinguish different biochemical process in the sulfur cycle. That is all I want to speak today. And thank you for your attention. And just let me know if you have any question or comments about this slide.